Hello YouTube, Mike Waldman here, Mushroom Rangers. Gonna do some terminology here for mushroom cultivation. Mycology, the study of fungi including genetic, biochemical, medicinal, and edibles. Mycologist is a microbiologist who studies and researches the microorganism fungi. Mycelium is the vegetative part of a fungus, a network of hyphae, the roots. AEE, like the roots of a tree or a tomato plant. Mushroom, or what they call toadstool, is the fleshy, spore-bearing fruit body of a fungus, i.e. it would be the same thing as an apple to a tree. So the mushroom is, i.e. the fruiting body of the mycelium. The spores, it's a tiny reproductive cell that allows fungus to replicate and grow, what they call fairy dust. Grain spawn, a sterilized grain inoculated with spores or a sterile culture of mycelium, which is called the seed. The substrate, material on or from which an organism lives, grows, or inhabits or obtains its nourishment. That's called the food. Agar, gelatinous substance obtained from seaweed used in biological culture media. So the life cycle is it the chicken or the egg? Where it goes. So you build the mycelium here, then it grows up, and then it grows into a mushroom. That's called pinning. And then it grows into a big mushroom, produces gills and a cap and a stipe. Now, when the gills release the spores fall back down to the ground and create more mycelium. So that's pretty much the life cycle. If you interrupt a mushroom before its life cycle, what will happen is the spores won't be able to disseminate and reintroduce the soil and start the cycle all over again. So more nomenclature is, you got a regular old scientific petri dish. This is a three-way. This is the petri dish on the dime. What's in there as far as the agar is the same as it is in here. It's just in a different vessel. And then you move from the petri dish to liquid culture, where it's active mycelial growth in satellite colonies. Then when you want to preserve your species, you go to an empty master slant tube, which this is it. Difference between this and this, or this, or this, is it has less nutrients, meaning that you're putting it into hibernation. So when you inoculate from your master to your master slant, it'll grow and colonize. As you can see here, Hen of the Woods, this is my master culture slant, meaning that this has as close to as possible the genetic material from when you cloned, transferred to a petri dish, and then into the master slant. And then you have your library here, which these are all master slants of different species. So now what I can do is, if I ever lose a strain, or if I find out that my strain is sort of lacking vigor what I can do is I can go back to my master slant tube, take a little piece of agar out of there, transfer it to a Petri on the dime, and then when it grows out successfully, contaminant free, then I'll transfer it to the master Petri dish. So over time, these will lose vigor, like maybe 2 years, 3 years. So about 1.5 years, what you're going to want to do is you're going to want to take a little piece of this, transfer it to this, transfer it to this, and then transfer it back to a blank master slant. That way it keeps propagating, you keep the genetics as close to its parents as possible, and then you can keep it going on. So that's what I'm doing now, is I am pressure cooking some grain spawn. What is grain spawn? Sterilized grain inoculated with spores or a sterile culture of mycelium. Basically the seeds. I'm basically making the seeds right now. So I can either inoculate it one of 2 ways. I could do the syringe with the liquid culture, which is faster, or I could take a piece of agar out and put it into there. Either way, it's the same process. However, with this, it's more sterile because I don't have to open this jar. I don't have to open the jars that I have because they're pretty much made the same way. They have this impervious, self-healable, self-closing port and gas exchange. Whereas if I use agar, then I have to open this up, cut it, open this up, and then put it in. So there's more chances of contamination. However, on the flip side, you will know that this is more sterile than this because it grows out white. You won't know if there is contamination in this unless when you make this, you test it out on this or this. Then you'll know it's 100% clean and clear. But then you also have to worry about vectors of contamination with your needles. So if you don't pressure cook them the correct way or clean them the correct way, that could be another vessel for contamination. Versus this, all I have is a scalpel where I flame sterilize it, rub it down with the isopropyl alcohol, cut a piece out, and put it in. Put it in the master grain spawn, which there's less of a transfer and less of contamination. So that is the breakdown. This is where I'm going with this video, or this series of videos, to show you how to start yourself up with stuff like this on a low key. Start making mushrooms and producing mushrooms for yourself, for your family, for your friends, and just to play around. So like, subscribe, tell your friends, tell yourself, keep it fungi, and blessed be.